How to do an ELISA. An enzyme-linked immunosorbent assay, or ELISA, is a method for detecting and quantifying the amount of an antigen using antibodies. This type of assay is frequently used in research and is among the most specific and accurate immunoassays available. There are several different types of ELISA assays. Each follows the same basic steps, but the order of antibody and antigen addition and the method of detection can vary. Let's look at each step of one type of ELISA called a sandwich ELISA. Sandwich ELISAs are commonly used in research and are especially good for detecting and quantifying antigens in sample types, like cell culture supernatant. First, a binding antibody specific to your antigen of interest is added to a plate well as a solution. The solution is incubated to allow the antibody to bind to the plastic wall of the well. Then the supernatant is removed, and the well is washed. Bound antibody is left behind. A buffer solution containing a mixture of proteins is added to the well. These proteins block non-specific binding sites on the antibody. Incubate and wash. Now, your prepared sample containing your antigen of interest is added to the well. Let the solution incubate. The antigen will bind to the specific binding site on the bound antibody. Wash. Now you have a surface-bound antibody-antigen complex. Next, add a solution of a second binding specific antibody. Incubate and wash. You have a bound complex of your antigen sandwiched between two specific antibodies. The next step is to detect this complex. Add a nonspecific binding antibody that is linked to a detection enzyme. This antibody will bind to the sandwich complex. Using a third antibody means you won't have to design a specific antibody-enzyme conjugate, a potentially time-consuming and expensive process. Incubate and wash. All that is left in the well now are antigen sandwich complexes bound with an antibody carrying a detection enzyme. The next step is to add a solution that contains substrate for the detection enzyme. The reaction between substrate and the enzyme will generate a color change or fluorescence. Incubate. Add a stopping reagent to halt the enzyme-substrate reaction. The concentration of target protein will determine the intensity of the color in the well. Now, use an instrument to record the absorbance of the assay sample. You can also quantify how much of your antigen of interest is present if you created a standard curve for your ELISA.